All right, so again, on this quick video here, it's just going to be, we're going to watch about the first 4 minutes of this video to recap what we've talked about so far with protein synthesis.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
All right. So we're going to stop here on that, and let's pick up where we left off on Monday. All right. So again, we're talking about protein synthesis here, and the essential question that we're understanding is, how is a protein made using the code of nucleotides from DNA? And this process is collectively referred to as the central dogma of biology, right? The central dogma of biology tells us that DNA, the information contained in our DNA, is used to make RNA, and that RNA becomes a code for the production of proteins. All right, so real quick, where does protein synthesis occur? Somebody tell me. Where does protein synthesis actually occur?
In the cytoplasm?
It does occur in the cytoplasm. Where specifically?
The ribosomes.
On ribosomes. And so where is this DNA located that we're using as our template for protein synthesis?
In the nucleus.
In the nucleus of
a eukaryote.
Of eukaryotes, right? So you have to specify with this answer. That's a very broad question. It has 2-part answer, right? DNA is located in the nucleus of a eukaryotic organism, but if it's bacteria, that DNA is located out in the cytoplasm. Thank you. All right, so we're here looking at DNA as the starting point, but we have to generate this RNA molecule, right, in order for us to continue the process. All right. So that first step of protein synthesis is called transcription, where we are synthesizing RNA molecules, right? Very briefly, you remember that RNA and DNA are two types of nucleic acids. These differ structurally and functionally, right? DNA is double-stranded, RNA is single-stranded. DNA has the AGTC basis. RNA does not have thymine, it has uracil instead, right? So there are some differences that we need to keep in mind about these two nucleic acids. Structurally, or from a chemical standpoint, RNA has the sugar ribose. DNA has the sugar what? What's the sugar found in DNA?
Deoxyribose.
Deoxyribose, absolutely. So if we looked at the chemical composition of that 5-carbon sugar, it's a pentose sugar, there's going to be an oxygen missing at that 2' carbon in DNA. And that's where this name deoxyribose comes from, because it's missing an oxygen. So there are a few differences that you should be familiar with in terms of the structure of these two nucleic acids, okay? Right, so RNA is single-stranded, has ribose sugar, uracil replaces thymine, okay? It can leave the nucleus with the DNA's instruction, but in prokaryotes, this process is gonna happen all out in the cytoplasm, okay? So make sure you have these differences or points of comparison in your notes. And I am pushing through kind of quickly because this information we talked about a little bit already, and we do have an exercise to complete before the end of class today. So I'm going to push through these real quick. We did talk about the 3 types of RNA already. We have our messenger RNA, ribosomal RNA, and transfer RNA. In your notes, you should have that these 3 messenger RNAs all have 3 different functions, right? So our mRNA is going to serve as the template, right, or the message for generating our protein, our polypeptide chain. The ribosomal RNA is going to be the major subunits of those ribosomes that are going to be responsible for translation, right? And then those transfer RNA, tRNAs, are going to function by bringing in the amino acids that correspond to those codons that we're reading from our messenger RNA, okay? Right, so the function of RNA is going to carries out DNA instructions by building proteins. So this RNA molecule is a very essential intermediate in the process of protein synthesis, right? If this first step, transcription, doesn't happen accordingly, then protein synthesis will be compromised, okay? We've got to have an essential first step of going from DNA to RNA in this process of protein synthesis, okay? So we already said that messenger RNA carries copies of instructions for building proteins, all right, from the nucleus to the ribosome. That's only true in eukaryotes, right? Again, in prokaryotes, this process is happening out here in the cytoplasm. We're going to get the transcription of DNA into RNA. And we know that we're going to have the complementary base pairing occurring, but instead of thymine, we have uracil in RNA, okay? Our RNA, we said that those are going to be in our structural components of the ribosomes. We've got our large and small subunits. You'll get some more information on that later. But right now we're understanding that these ribosomes are an essential piece of the puzzle because this is where actual translation takes place, okay? This is where those tRNA molecules are bringing in the corresponding amino acids based on those codons. So that anticodon, all right? Transfer RNA, again, the function here is to transfer an amino acid to the ribosome to be added to that peptide, to that protein that we're building, right? So there is a tRNA for every amino acid that's being brought into this chain, all right? So the amino acid is attached to this transfer RNA and it's being brought over to this ribosomal complex, and that's where translation is occurring. All right. So a great concept check is for you to be able to list 3 types of RNA and their functions. All right, we're going to keep going. So I mentioned this phrase, central dogma of biology. This is the standard or the postulate that we understand for how we go from the information that's contained in our DNA to proteins. Okay, write that down. You will continue to hear this reference to the central dogma of biology. Okay, it is how we go from DNA to RNA to protein. That is the direction of information flow, where the information contained in our DNA, in our genomes, serve as a blueprint for the production of proteins, right? And we know that proteins are the ultimate molecules responsible for cellular activity. That's why we care about this. Okay, these proteins become the structural and functional components of the cell, right? Which is the basic unit of life. So it is absolutely imperative that you understand the mechanism by which we make these proteins. Okay, so we're clear that protein synthesis occurs in 2 steps, right? We talked about this on Monday. We've got transcription and we've got translation. You're going to spend as much time as you need to out of class understanding what's happening in these 2 processes. Okay, very simply put, during transcription, DNA is transcribed into RNA, and that messenger RNA that is made becomes the message for the second phase of this process, which is translation. Translation is when we're now decoding, right, that sequence of messenger RNA. We're reading it in groups of three nucleotides, right? And that is called a codon. So we decode the codons, and the transfer RNA will continue to bring in the amino acids that correspond to those codons. And that is how we synthesize a protein chain. So again, I'm just kind of repeating myself here. Protein synthesis in prokaryotes. So you've got all of this activity taking place at the same time in the cytoplasm. Replication, transcription, and translation all occur in the cytoplasm of prokaryotes. Okay, we're very clear about that. In eukaryotes, this process are separated by time and when it actually happens. Okay, replication and transcription takes place in the nucleus of eukaryotes, and the mRNA that is generated in the nucleus will be transported out of the nucleus and into the cytoplasm, where it has access to ribosomes for the second part of the process. Translation occurs. Okay, translation occurs on the ribosomes, and we've got this growing polypeptide chain where our transfer RNAs are bringing in our corresponding amino acids. So in terms of description, we're saying that transcription is the process of copying a specific portion of a gene, producing a complementary strand of messenger RNA. So we start with the DNA sequence, right? And we're going to generate a complementary messenger RNA, right? So this complementary base pairing, we have to know the rules for base pairing. A pairs with T, G pairs with C, right? But with messenger RNA, the T's are replaced by what? What nucleotide base, which nitrogenous base is going to be found in messenger RNA that's not found in DNA? Uracil. Uracil, right? So as we're as transcription is taking place, we're synthesizing this messenger RNA, and we're synthesizing this strand with the complementary base pairs, or the complementary bases to the strand of DNA that we start with. Okay, so make sure you understand that process. All right, and so what we're saying here functionally is that this messenger RNA acts as an intermediate, okay, between the DNA and the protein synthesis. Okay, so RNA is an intermediate molecule in this process. Okay, so yeah, so you can think of it as a temporary copy. You can think of mRNA as a temporary copy of DNA that is used, and then it's no longer useful. So that messenger RNA will be ultimately translated into a growing polypeptide chain. Now, here's the point that we kind of left off on Tuesday. Like, how does this happen? This RNA polymerase, write this down. RNA polymerase is the enzyme that's responsible for this process of transcription. Okay, so the RNA polymerase, which is an enzyme, is going to attach to a very specific site in our DNA sequence that we'll call the promoter region. Okay, it's part of this DNA, and it's going to tell us where we're going to start this process of transcription. All right, so we've got RNA polymerase here. RNA polymerase uses one of the DNA strands again as a template to generate this new complementary strand of messenger RNA. Okay. DNA becomes a template for the synthesis of RNA. Okay. Again, we're following our general base pairing rules for that you learned about in DNA replication, except in this case, we're going to have thymine replaced by uracil. All right. These are the complementary base pairings for replication and transcription. All right. G pairs with C, C pairs with G, A pairs with U when we're generating RNA, and T pairs with A. All right. So DNA bases, we have our A, T, Gs, and Cs. When we're generating our messenger RNA, A will be pairing with U in our newly synthesized messenger RNA. All right, so we're clear about what happens with transcription. DNA is transcribed into RNA. All right, so we know what happens there. So again, the last piece of that puzzle here is that the messenger RNA now has to be translated. Okay, the second part of protein synthesis is translation. Translation is when we convert that messenger RNA into an actual protein, right? This polypeptide chain. Transfer RNAs are going to bring in, okay, the corresponding amino acid based on the triplet codon we're reading here, right? We're reading these messenger RNAs in groups of 3, right? Those codons, and they all correspond to an amino acid. We looked at the genetic code on Monday, and we understand how to read the genetic code in groups of 3. And there's a corresponding amino acid that will be brought into this growing polypeptide chain by way of those transfer RNAs. Okay, so we'll stop here for the purpose of this course. There's a little bit more details that kind of go into this process that you'll continue to explore in some of your higher level courses. But I want us to not lose sight of the big picture here. I'm going to stop my screen share for a second because I do have an exercise that I want you guys to complete before you leave today. So we have about 25 minutes remaining. I'm going to give you guys the rest of class. I'm going to put you in breakout groups. If you look in your Blackboard course, you know the routine here. This is a group exercise. You guys are going to go into breakout rooms and you're going to respond to these concept review questions collectively. You should have all of the information in your notes. They're very short concept review questions. I think there are 7. You're going to work in a group. You're going to designate one recorder and one timekeeper. Okay. Your recorder is going to jot down the answers for all of the group. Okay. You're going to submit one page with everyone's name on it. And the timekeeper is going to help you stay up on the time. So I'm going to give you breakout rooms. Head over to Blackboard now, and under content, you will see protein synthesis review. Okay. I'm opening your breakout rooms now. You have been, oh, wait a second. One second.